Thank you, Christina, for the introduction. So as Christina mentioned, I will be discussing Peaks Online 13, which is our proteomic solution with high sensitivity, accuracy, and speed. So to start off, for those of you who may not be fully familiar with BSI products, we do offer a whole software suite for proteomic solutions, as well as antibody sequencing and glycoproteomics. We also just recently released Proteoform X, which is our solution for proteoform characterization. Now, although we have a lot of software services, we also do have a very comprehensive lab service for antibody sequencing, immunopeptidomics, glycoproteomics, as well as just general proteomics too. However, for this presentation, I will be mainly focusing on Peaks Online, which is our solution for high throughput multi-user software. Now, I'd like to introduce Peaks and how it supports comprehensive bottom-up proteomic workflows. And actually, Peaks Studio and Peaks Online are really built on the same core algorithmic logic. That means whether you're working on Peaks Online or Peaks Studio, you're using the same high-performance de novo-driven analysis framework. And Peaks Studio provides the complete bottom-up proteomic solution to handle both discovery, so DDA and DIA data, as well as our targeted solution as well for PRM and hybrid DIA. Now, for Peaks Online, we are mainly focused on discovery proteomics, so mainly our DDA and DIA workflows. However, we hope to expand in the future into our targeted solutions. So we are a vendor-neutral software, so regardless of what mass spec vendor you use, you can load in the raw data into Peaks and analyze it without actually having to convert the data. And for each data type, we have a workflow-driven analysis for each of the types of acquisitions. We also do support ion mobility as well. So if you have an instrument containing ion mobility, we do support that to allow you to load the data into Peaks. Now we have many variable flexible workflows, and really de novo sequencing is what really powers Peaks, and it enables the identification beyond database search constraints. So this allows us to have some pretty cool specialty applications, such as searching for PTMs or sequence variants, as well as we do also provide quantification tools supporting both DDA and DIA datasets. Now for Peaks Online, it is our server-based solution, and it is also multi-user as well as scalable. So a bit of an outline for today's webinar. I'm going to discuss Peaks Online architecture. I'll also get into a little bit of our streamlined workflows, mainly focusing on DIA and DDA. However, I will be focusing more on the DIA solution and just touching a little bit on our DDA solutions. I will finish with a software demo, and then we will have a live Q&A, so if you have any questions, feel free to ask then. So let's start specifically about Peaks Online and the advantages of a server-based platform. So this is kind of an example of the web browser. So when you first go to log into Peaks Online, this is what you'll be displayed. And everything is actually deployed on the user's end for full confidentiality. And Peaks Online is really designed to take advantage of powerful and shared computing resources to perform these peptide and protein identifications. So the platform has been completely restructured to support high efficiency processing at scale. Large data sets can be run and analyzed more quickly. And most importantly, we allow multiple users to run projects simultaneously without compromising the performance. This really makes it valuable for core facilities, collaborative labs, and high throughput environments. So Peaks Online actually offers two different interfaces. So we have the web client interface and we have the command line interface, or CLI. And the web client interface provides a graphical user interface designed for interactive workflow configurations and in-depth data exploration. So this essentially happens through a standalone web browser. So you can easily set up your workflows, adjust parameters, or monitor job progress all through a web browser such as Chrome or Edge, which makes it really easy access for you. The CLI, or the command line interface, is really designed for automation, so it allows the user to automate data analysis workflows as well as results exporting, making it really easy to integrate peaks into existing pipelines. So the user will essentially define an analysis parameters in a text configuration file, and Peaks Online will actually automatically retrieve the data from a particular drive, run the analysis based off those predefined parameters, and you can set specific exports to be set for whatever the result you may be looking for. And this really enables fully automated hands-off data processing for a high throughput environment, reducing manual intervention. So to support scalable and efficient data processing, Peaks Online is built on a distributed architecture consisting of 3 core components. So the first I'll be talking about is the master node. And the master node really acts as the central point of coordination within the system. And it serves as the main point of contact for users and manages the web-based interface. Essentially, the master node is responsible for scheduling tasks, synchronizing computational workflows, and as well as distributing those analyses across those available resources. Next, we have our worker nodes. And the worker nodes are the primary processing units that perform all of the analyses. And these really work for data processing and computation, and they can be configured to use a custom number of threads. And typically we suggest having 2 or more worker nodes to have Peaks Online function to its best ability. Next, we have the database nodes. And the database nodes store application data and provide data access to the master and worker nodes. And this whole system really ensures that Peaks Online can handle large data sets as well as multiple users while maintaining the required stability. So as I mentioned, Peaks Online is really designed for large-scale projects, and I did mention that worker and database nodes can be easily added for both vertical and horizontal scalability. So I have a figure here, and this is a little bit outdated as more resources are becoming required on the software side, and the size of the data is increasing, as well as the number of data files users are looking to analyze. So we now actually start our licenses at 128 threads for Peaks Online. For example, Studio starts at 24 and 48 threads. So we really require a lot of computing power for Peaks Online to be able to perform these analyses. I also just wanted to mention the use of a GPU. So a GPU can be used to essentially boost your speed, and we really recommend it for particularly the DIA Peptidome and DIA database search workflows. We do have the specific specifications here for that GPU, but if you're ever unsure, please do reach out to us and we can always help you with the requirements for Peaks Online. So to ensure transparency, performance tracking and administrative controls were added into Peaks Online. So we have several monitoring and management tools. So the first is the worker monitor, and the worker monitor allows administrators to observe queued tasks and monitor communication between nodes. And this will essentially show you which analyses are running or waiting in the queue. And next we have the master monitor, and the master monitor will allow you to monitor memory consumption and overall resource usage, checking on the stability and responsiveness of the system. The manage users function allows the user to control either user or the administration accounts, and you can add new users or remove them. This is really where you can manage all of that. And lastly, we have the project summary view. And this is where you can see all projects that have been run on the system, as well as completion state, and users can quickly access these running analyses, review completed projects, and monitor overall activity. So I just talked to you about the redesigned architecture for Peaks Online. I'm going to mention to you a little bit about our streamlined workflows, as well as mainly discussing DIA Proteome, in which we've actually improved the speed and performance quite a bit, as well as also providing the user the ability to search for mutations, mainly sequence variants, and results transparency. We've also done a lot of changes to the DIA Peptidome workflow in particular, where we have also allowed the user to search for sequence variants, as well as more peptide-focused searches. So that includes searching for peptide candidates in a peptide list in the form of a FASTA file, as well as through a spectral library and still our database searches. So jumping into the streamlined workflows, so Peaks Online shares the same algorithms as Peak Studio, except we've just optimized it to specifically handle large-scale studies. So users can actually customize their analysis workflow, selecting which steps to run and adding or modifying the steps really at any point. So we do have two main application-driven workflows. We have our Proteome workflow, which is essentially searching for what proteins are found in the sample. And we have our Peptidome workflow, which is searching for what peptides are found in the sample. And we have many comprehensive steps with specific specialized algorithms for each step. As well as, I'd like to say, we are very flexible and user-friendly, so you can actually pick your steps really as needed. So if you want to perform a database search and then maybe later you want to modify and add an LFQ, you can always go back to your previous ran analysis and add another analysis to it later. So first we have our DDA workflows. We have our Proteome and Peptidome. So the Proteome workflow typically begins with de novo sequencing, followed by our database search. We also provide the user to search for all variable PTMs using Peaks PTM, as well as mutations using Spider. And any of these peptides can go along to be quantified, as well as proteins too. Next, we have the Peptidome workflow, and this is really a specialty workflow. So it looks quite simple here, but there's actually a lot going on in the background because it's integrated with deep learning-based de novo sequencing, as well as canonical and non-canonical database searches. And we also use Spider in this workflow to allow you to look for mutations. For DIA, the Proteome workflow is a little bit different. So for DIA, as more unknowns need to be identified, we typically start with a library search. So some spectra can be eliminated, and then we go on to the database search. However, a library search is not necessarily required. You can always just start with a database search. So if you don't have a library, you can still perform the DIA Proteome workflow. And after the database, de novo sequencing can be done, and we can search for mutations using Spider, as well as identify novel peptides. And we also have quantification supported in this workflow as well. For deep novo peptidome, again, it does look very simple, but it is a bit more complex as it is deep learning based. And we've also added the ability for candidate searches now that we have the peptide lists as a FASTA file or a spectral library allowed to be searched. So let's talk a bit more detail about the DIA Proteome workflow and updates to this workflow. So the first really being performance. So we've really improved the speeds. Just on the graph on the right here, this is our real-time data analysis and the time it took. And with more and larger data, this is really the focus for many researchers is the speed of the analysis. And here we are comparing the running times between Peak 13 and a third-party competitor. However, for comparing our algorithms, we do use Studio to easily match threads to the third party and Studio. However, Studio and Peaks Online are built on the same algorithm, so this is typically why we use Studio to do that kind of benchmarking. So not only are the speeds increased, we've also increased the performance of ID and quantification, as well as reproducibility, and it's really highly competitive, as shown here, and we're always looking to improve. So I really like this slide because it's really a quick screen of what you're expecting in the data for DIA Proteome. And we really incorporate 4 different methods of peptide identification. So we have our spectral library search, database search, de novo sequencing, and sequence variants. And the search is really performed using an expanding search space for improved coverage and increased FDR control. So first, we search against the spectral library of previously identified spectra. However, as I mentioned, this step is optional, so you can directly perform the database search instead if you do not have a spectral library. And by estimating the FDR, peptides that pass this filter are saved, and any MSMS spectra that don't match a peptide with the FDR are brought forward to the database search. So if it passes the FDR, it will be reported as a library peptide. If not, it will go on to the database search. And any other unmatched spectra that are analyzed using de novo sequencing, those peptides, if they pass the ALC score or the average local confidence score, they will further be subjected to the SPIDER algorithm. And what's not matched with SPIDER based off mutation and ion intensity, they will be passed along with ALC, or a high confidence sequencing score will be reported as for the novel peptide. And if it passes that ion intensity, it will be reported as a sequence variant. And I'd just like to mention for the DIA proteome workflow, the library and database peptides can go along for LFQ. So a little bit more mention about SPIDER. So de novo sequencing has been used with sequence variants in DIA to provide a deeper discovery for you. And this really is unique to Peaks for DIA to be able to look for sequence variants. We have a de novo sequencing algorithm for DIA, and we particularly adapted SPIDER to DIA to be able to find sequence variants. So essentially what we'll do is we'll compare the de novo sequence against the database sequence, and we will then try to permutate, and if it matches that MS2 information, we'll be able to construct the real sequence based off both the de novo and the database sequence. And I think that this is really cool, as well as you can easily see this in Peaks Online and Studio as well, the comparison between the de novo and the database. So now that the architecture is more stable, we're actually able to add a lot more figures to the user interface for more comprehensive results reporting similar to Studio. And the idea is really to allow the user to validate the result that they are getting and investigate what we match at the fragment ion level. And if it doesn't match, please do complain to us. We're always happy to help and take your feedback so we can improve. And this is why we've really given you the chance to investigate and confirm your result. One other thing I would like to mention is a spectral library. So you can actually create a spectral library from any previous analysis on Peaks Online. And typically, of course, DDA data is perhaps fractionated to form a spectral library, but you can also create a spectral library from DIA data or de novo sequencing. And I think that it's cool to have this in the software so that you can do it all in one platform. So you can create your spectral library and you can use that spectral library to search in your DIA data. However, if you do have another spectral library, you can upload that in another compatible format. So next I'm going to talk about DIA Peptidome and the new improvements to that. So DIA Deep Novo Peptidome has specifically been optimized for both accuracy and speed, and we now support a user-defined candidate list in the form of a spectral library or a peptide list containing peptides expected to be found in the samples. And this figure here is actually quite simplified, but the algorithm follows really the same logic as classical DIA in terms of step searches for improved FDR and increased coverage. So here you will be able to assign your spectral library and peptide list, and either or these will be reported as candidate peptides. Then we will have database peptides, as well as we do perform Spider as well to search for homolog peptides, and anything that does not match but still has a high ALC score will be reported as a deep novo peptide. And this is kind of what this looks like in Peaks Online. So you can set your spectral library or your target peptide list, and of course your database. And what this actually looks like in the result is they will be reported as candidate peptides if it was searched as a peptide list or spectral library. And you'll have your database as well as mutations and your deep novo peptides. And in case you want to investigate further, in the peptide tab, if you look at a particular peptide, and if you sometimes have to scroll all the way to the right, you can see that there's a found by column, and that will actually be indicated where that particular peptide was matched to, so if it was matched to the candidate list or the database search. So I'm just going to quickly mention a little bit about DIA Peptidome and Proteome. And if you have DDA data, the immunopeptidomic solution is actually quite elaborate in Peaks, and it's a really complex algorithm that includes both a first-round search using a canonical database, and as well as a second-round search using a non-canonical database. And we do perform Spider to search for any mutations in the sample as well. And any unmatched spectra will once again be reported as deep novo peptides with a high score. And any of these peptides can actually go along to perform label-free quantification. And I guess really the whole idea is we also have this workflow that is designed to bridge the gap between proteomics and genomics, where we've actually integrated a comprehensive gene table into this workflow. And this gene table will map peptides to their genomic origins to visualize the origin of the peptides, directly linking them back to their corresponding gene. So we're able to provide you gene site information, as well as the overlap between canonical and non-canonical peptides, as well as any deep novo peptides that maybe there was an amino acid tag will also be shown here. And typically when it comes to proteomics, the aim of most studies is to identify a peptide and then typically a protein. So for particularly DDA proteome, and really any workflow, Peaks starts with feature detection and then MS2 association. However, for a DDA workflow, we actually begin with de novo sequencing. And it's really exclusive to Peaks is that we take advantage of this to incorporate these for an increased confidence in database matching. So Peaks will do de novo sequencing, and this will result then will be used for some tag searching for protein shortlisting, which will allow for an improved search space when we move on to peptide spectrum matching. And in the identification result for the database search, we'll report a PSM score. And the PSM score is scored based off many parameters, including the similarity to the de novo derived sequence. And it will essentially depend on the similarity of the database to the de novo sequence. And in addition, any of those good scoring de novo tags we actually use to search for PTMs and as well as mutations. So I hope I was able to kind of show you how Peaks Online is pretty high throughput, multi-user, secure and scalable, as well as I did mention a little bit about our deep proteome and peptidome workflows and our unique deep learning based algorithms. Now I am going to jump into the demo. Let me just swap screens here. All right, so when you first log into Peaks Online, you will be greeted with the project summary page. And as I mentioned on the project summary page, we have all the projects that have been ran. You can see the name of the project, what time it was created, the number of MS runs, as well as the number of analyses. You can also see the status of the project too. And we do have many different configurations here. In Peak Studio, this is called configurations. Peaks Online, we just call it settings, where you can add different databases, peptide lists, or spectral libraries, as well as you can configure any modifications or enzymes here. So if you essentially click on one of them, you'll be taken, for example, to the managed database list. We also have an admin tab here if you're logged in as the administrator, where you can check on the master, worker, and the data node, as well as the users. Again, information about your account. And lastly, we have our help tab. So I just wanted to quickly take you through a demonstration of what setting up an analysis in Peaks Online kind of looks like. So if you click this Create New Analysis button, you'll be taken to a page where you can name the project. And I'm going to quickly just go through DIA Proteome. So you can select a new workflow, or you can use a previous analyzed workflow that you want to use, or just create a new one. So today I'm going to be creating a new one, and I'll just click Next. And you can upload the data to Peaks Online either locally or remotely, and easily add it here. One thing some users don't actually know about is the ability to name samples based off delimiter. So if you have named your raw data file in a specific way and you want to separate the naming, for example, if I click underscore, and maybe I just want to name my files based off something that was already written in the original name, I can easily just select that, and it will automatically name based off the delimiter I choose. We also have many built-in enzymes, so you can always customize your own enzyme or use any built-in ones here. We have many different types of fragmentation methods too, so always select the fragmentation method that was used for your analyses. As well as we have a list of many different instruments. If you want to customize your own instrument, we do recommend discussing with us, just because we have specific algorithms towards these instruments listed here. So always reach out to us if you have maybe an instrument that's not listed here and you want to load it into Peaks. We're always happy to work with you on that. Lastly, you'll select your acquisition type, and for Peaks Online, we have DDA and DIA. So I'm going to click Next, and we have our proteome and deep novo peptidome workflows. So I'll click proteome. So first, you can set your error tolerance, so you can manually set the error tolerance or check auto detect. We also have match between runs. We only recommend using match between runs if your identifications are expected to be similar in a specific sample. We also have the ability to add a spectral library, as previously mentioned. So you can add a library maybe you have already uploaded, or you can easily click this plus button and you can create a library based off a previous analysis. Protein inference will automatically use whatever database you set in the database search parameters. However, if you wanted to just perform a spectral library search, you can set that database here as well. And if I turn the database search back on, it will automatically actually be set in the database search parameters as well. So you can set your enzyme, however we already set the enzyme, and we have our digest modes as well as miscleavages. You can set whatever you want to search for. You can also set the PTMs you wish to look for as well. And typically I recommend using up to 4 variable modifications. This is just simply because using more variable modifications will expand your search space, so it's always good to be mindful of that in terms of your analysis. You can select whatever database you would like, depending on your sample, as well as you can add a contaminant database to search for. We have many different filters here for the database search. If you have a particular peptide length you want to search for, or a charge, you can set that easily here. And to find mutations with sequence variants, you're just going to slide this button right here, and it will automatically turn on the de novo sequencing in order for Spider to run. If you wanted to add quantification specifically for DIA, we have label-free quantification, so you can easily set your groups. So for example, if I was searching control groups versus treated, I would set it like this. And you can easily change the color or the name of these groups and set your base sample as well. So on the next page, we have our report filters and our LFQ filters. So you can search based off the protein group FDR or -10 log P. And for the LFQ report filters, we always recommend using kind of what's set here by default. However, you can always go back and change and easily modify the analysis. I want to just point out for our normalization methods, we have use internal standard proteins here. You can see right now that this is kind of grayed out. This is because you have to perform a database search first before you can actually perform the internal standard proteins, because we need to have the identifications first before you can select those internal standard proteins. I would just hit submit next to analyze the data. However, I am just going to go into already ran data to kind of show you essentially what the results would look like. So on the results page, the first note here is whatever you essentially named your analysis as, and you're given the analysis properties. So whatever you had previously searched your analysis as. And as I mentioned, there is this modify parameters button here up in the top right corner. So if I actually click that, it will take me back to everything I previously set in that particular analysis. And I can just go back and change whatever parameter I want if I wanted to rerun something. And I just want to point out that now that use internal standard proteins is now there. Now that there is identifications, you can now set. So you will just have to run in order to normalize to that. So the first node here is the data node. And in the data node, this is kind of a summary of all the samples you ran, what you loaded and ran the data as, so what enzyme you used, fragmentation you used, the instrument as well. One thing I want to point out is if you click this drop down, you'll be able to then view the raw data if you go to this view fraction button. And this takes you to the data where you can investigate more on your MS1 or MS2 data. The next node in the list is the database search. So typically any peak search, regardless of the workflow, has a summary tab, which is just a summary of the data statistics of the analysis that you just ran. So for example, we have our summary statistics table, which is like a summary of the different peptides and proteins found. We always have the false discovery rate curve, as well as some other plots about mass error and retention time calibration. We do also provide a PTM profile in the summary, as well as the number of miscleavages. We also provide those statistics as well. The next tab is the protein tab, and the protein tab essentially is a summary of all proteins found in the sample. We also have our coverage view down here, where if you want to look at the spectra for a particular matched peptide, you can always pull that up and check on that spectra. We also have our protein filter button, so you can filter by whatever modification you want. Even if there's a protein name you want to search for or a description, you can easily do this and then hit apply. And that will essentially change the search or the filter based off whatever you set here. And I want to mention that if you want to export the results of that particular filter, you can easily click this button in the top right here, which will export whatever is displayed in this table. And this goes for the peptide and the sequence variant, as well as the novel peptide tab as well. Next, we have the peptide tab, which is a summary of all the peptides that were found in the search. And again, we have now provided a lot more information about those particular peptides, including MS2, XICs, as well as precursor and survey scans. The sequence variants tab will be a summary of any sequence variants found in the sample, as well as the novel peptides tab are going to be any de novo peptides, de novo peptides found in this case that have a good high ALC score but were not matched to the database. So with that, I hope I was able to kind of show you what Peaks Online can do, and I will be happy to take any questions.